Hello, everyone, and welcome to Lecture 14 on States of Matter and Colligative Properties. We first need to recap a little bit about states of matter and changes of state, specifically, where one substance is transformed from one physical state to another physical state. This means that no new substance is going to form. Only the chemical composition, the physical state of the chemical composition will change. The chemical composition remains exactly the same. We're going to talk about 6 possible changes of state: freezing, melting, evaporation, condensation, sublimation, and deposition. Now, some of these you're probably very familiar with, and some of these you may not have heard about. Freezing, of course, is a change in physical state from liquid to solid. Melting, from solid to liquid. And evaporation, from a liquid to a gaseous state. Condensation is the change from a gaseous state back to a liquid state. Sublimation goes directly from a solid to a gaseous state. It does not go through the liquid state to get to the gaseous state. And deposition goes directly from a gaseous state to a solid state, not going through the liquid state to get to the solid state. These processes are associated with thermal energy, either the absorption of thermal energy, which is referred to as an endothermic process, or the release of thermal energy, which is referred to as an exothermic process. Endothermic processes include melting, sublimation, and evaporation. Now, with melting, we're going from a solid phase to a liquid phase. And you might be familiar with this if you've ever dropped an ice cream on the sidewalk in Southern California in the summertime. There's no 5-second rule. You're not going to pick that up and start eating it. Well, hopefully you wouldn't. Because as soon as that ice cream hits the 100+ degree sidewalk, it's going to start melting. It's going to start going from a solid to a liquid phase. Sublimation, we go from a solid phase directly to a gaseous phase. And they're very familiar with sublimation on the East Coast, because on the East Coast it gets really cold in the winter, and so people wear a lot of wool clothing, which is very expensive. They don't wear it, however, in the spring and summer. They have to store it. And there's a little moth that is like a parasite to wool clothing. It'll eat little holes into that wool. And so people store their wool clothing in closets with naphthalene, or what's commonly known as mothballs. Naphthalene sublimes from a solid directly to a gaseous state and repels those little moths so that the clothing isn't ruined during the spring and the summer months when it's in storage. You're probably familiar, especially with Halloween being recent, with dry ice subliming, going directly from a solid state to a gaseous state. With evaporation, go from a liquid state to a gaseous state. Evaporation occurs at the surface of a liquid, where the particles have gained enough kinetic energy to escape the liquid and go into the gaseous phase. In order to gain that kinetic energy, those particles have to have absorbed some thermal energy. Exothermic processes, on the other hand, release thermal energy. So freezing, for example, where you go from a liquid state to a solid state. If you put liquid in an ice tray and put it in the freezer, that liquid has to release energy in order for the particles to slow down enough that they can freeze and form a solid. Condensation is going from the gaseous state to the liquid state. That means that the gaseous particles, which are widely separated, have to come together. And in order to come together, they're going to have to release energy so they can slow down enough, again, to form the liquid state, which is more condensed than the gaseous state. In deposition, we go directly from a gaseous state to a solid state. In this case, you could think of snow, for example. Snow or even hail is formed from deposition. It goes from the gaseous state in the clouds directly to a solid state and falls as snowflakes or as hail. Evaporation is a process by which molecules escape essentially from the liquid phase to the gaseous phase. And again, they have to have enough energy to do that, and this happens at the surface of a liquid. There are differences in the kinetic energy between the molecules that are in the liquid, and constant changes in kinetic energy of every given molecule that's in that liquid. This results in collisions with neighboring molecules, and those collisions with neighboring molecules help to increase the kinetic energy. There's a surface phenomena where molecules escape from the liquid phase to the gaseous phase, and that surface phenomena is called surface tension. The molecules at the surface have fewer attractive forces, because they're only attracted to the molecules in the liquid that are below them since they're at the surface. They're not attracted to the gaseous molecules that are above them, and the gaseous molecules that are above the surface molecules in the liquid are very, very far apart because gaseous molecules have a lot of space between them. On the other hand, those molecules that are at the surface have a greater potential energy. They're not completely surrounded by other molecules that are in the liquid. Again, they're only surrounded by molecules below them in the liquid. Greater surface area results in increased evaporation, but greater volume does not necessarily result in increased evaporation, provided you have the same surface area. So again, evaporation occurs at the surface of the liquid. So greater surface area will increase the rate of evaporation, but given the same surface area, if you increase the volume, that will not increase the rate of evaporation. These two videos that I have at the very top of this screen are very, very helpful. So this one is, the bottom one is particularly helpful in describing what I'm going to describe to you on this screen, and has a couple more simulations that will help you to understand and to see the movement of the particles rather than something static like I have on this screen. This top video here, though, is very, very interesting. It's not very long, and it's really well worth watching. It's a slow motion simulation that they show you where they drop milk into water, and essentially you can see the elasticity of the water from the surface tension. Very, very, very interesting and very quick. It's not a long watch, so it's well worth your time. So let's talk about the surface molecules having fewer attractive forces. Let's look at that first. So if we look at this diagram that I have, you notice that this molecule that's in the body of the liquid over here is completely surrounded. And even though this is a 2D shot, you can imagine that that molecule is surrounded on all sides, three-dimensionally, by other molecules. And all the little blue spheres are representing molecules. So there are attractive forces to these other molecules in all directions. And we haven't learned about something called intermolecular attractant forces. But intermolecular attractive forces are not covalent bonds, they're not ionic bonds, but they are sometimes called bonds, particularly when we're talking about something like water. They call them hydrogen bonds. Again, they're not covalent or ionic bonds, so they're nowhere near that strong. They're an attractive force. They're very weak, but still they are additive. So just like your spine has very weak joints at every vertebra, but those joints are additive, so that increases the strength of the spine such that you can sit upright and you can stand up. These sorts of attractive forces are similar to that. Again, all of these molecules that are in the body of this liquid are going to have those attractive forces to all of the molecules surrounding them. But when these molecules are at the surface, such as this one that they're pointing out, or any of them that are at the surface for that matter, they only have attractive forces to those like molecules that are below them. And remember, what's above the surface of this liquid is the vapor or gases, and gas particles are much farther apart than liquid particles. So the liquid molecules don't have attractive forces to those gaseous particles that are in the vapor phase. But they do have attractive forces to those molecules that are below them. So what's interesting about this phenomena is that it means the net pull is going to be downward here. So the net pull on this molecule is going to be straight down, while the net force on any of the other molecules that are in the solution is going to be equal and opposite surrounding it in all directions. Because that net pull on the surface molecules is either straight down or to the center if you have a droplet, that causes a surface tension because the force pulling down is so strong from all sides. And so you can see that you have a force pulling down here and a force pulling down here. Neither one of these forces to the side pull up, and so they're still keeping this molecule right where it's at on the surface. Those forces increase the water molecule's energy at the surface. Another issue has increased the water molecule's energy at the surface. Now, I want you to picture a diver standing at the bottom of a high dive ladder. While the diver is standing at the bottom of the high dive ladder, his potential energy is constant. Just like a molecule that is sitting way down here at the bottom of this liquid. Potential energy is constant. But as the diver climbs the ladder, the diver's potential energy increases and increases and increases. As this water molecule that I have a green arrow pointing at climbs to the surface, it has to disrupt the attractions of all of the molecules that are surrounding it. As it disrupts those attractions, it's moving towards the surface against a force. When you move against a force, when you move any distance against a force, in chemistry or in physics, that is called work. When you move a distance against a force and do work, that increases potential energy. So just like the diver climbing up the ladder, he's moving a distance against the force of gravity. And so he's gaining potential energy. When he gets to the top of the ladder and walks out to the end of the high dive, he has a substantial amount of potential energy. Just like when this particular molecule at the bottom moves a distance that is to the surface against the force of the attractions of the other molecules that are trying to hold it down in the solution. When it gets to the top, it has gained potential energy. So these particles at the top may have fewer attractive forces because they're not surrounded by other like molecules, but they have greater potential energy. And because the forces are all pulling in one direction, that means that the energy force pulling down or pulling towards the center of a droplet is greater. All of these things contribute to the surface tension. Evaporation occurs at the surface of a liquid. So which container here has a greater rate of evaporation given equal volume? If both the container on the left and the right have an equal volume, then the container on the left has a larger surface area. And so the container on the left is going to have a greater rate of evaporation than the container on the right that has a smaller surface area. But on the other hand, what if the glassware on the right has a larger volume? So suppose that we increase the volume in this glassware relative to the volume that's on the left. What will happen to the rate of evaporation? Will it increase? Will that cause both of them to have an equal rate of evaporation? Because now we've increased the volume? And the answer to that is no. Because remember, again, evaporation takes place at the surface. So increasing the volume and still having a small surface area relatively is not going to increase the rate of evaporation. Temperature affects evaporation, though. If we increase the temperature, we increase the kinetic energy because kinetic energy is directly related to movement. And if any of you have ever boiled water over a Bunsen burner or even on the stove, you see that you turn the heat on to the pot of water, you walk away, when you come back, everything's moving around. Everything's moving around because the increased temperature has caused an increase in kinetic energy. You increase, therefore, the rate of evaporation, because if you've increased the rate of kinetic energy, then you increase the ability of molecules to escape from the liquid phase into the gaseous phase at the surface. Those escaping molecules are collectively referred to as vapor. Vapor is the gas that exists at a temperature and pressure in which it would typically be considered a liquid or a solid. So when we talk about evaporation and increased temperature, we're not so much talking about putting something over a Bunsen burner and heating it up. We're talking about maybe the room temperature is 22 degrees C, or maybe the room temperature is 25 degrees C. Those are the kinds of things we're talking about, whatever the room temperature is at atmospheric pressure. When we talk about putting something over a Bunsen burner or over on the stove and heating it up, now we're referring to boiling, and that's a little bit different kind of evaporation. What are the effects of increased temperature then? Again, increased room temperature, and so maybe 22, 23, 24, 25 degrees C. What are the effects of increased room temperature on evaporation? Well, we can see that the liquid volume will decrease. That's clear because when we see, all of us have seen that water can evaporate, whether it's out of a glass, whether it's out of a pool, whether it's out of a lake. We can see that water will evaporate when there's a difference in the room temperature, the ambient temperature that's surrounding it. What we don't really see and what we don't really measure is that the liquid temperature decreases. So we can see the volume decreasing, but we can't so much see the liquid temperature decreasing. And we don't normally measure that. If we did measure it, it would be only slight differences, but there are differences. And why would the liquid temperature decrease as evaporation occurs? And this is under any circumstance. When you have evaporation, you not only have the volume decreasing, but the temperature also decreases. As the molecules at the surface that have high kinetic energy escape, then the kinetic energy that's associated with the volume itself is losing molecules. Less and less molecules means less and less kinetic energy because they're not bumping into each other and colliding with each other as much because there aren't as many of them. And so they're not generating as much kinetic energy. Kinetic energy corresponds directly to temperature. So as you decrease the kinetic energy, you also decrease the temperature. And this diagram is just further explaining that. So you can see that we have liquid here, and then we have vapor up at the top here. These molecules are going from the liquid phase into the vapor phase by evaporation. As those high energy molecules leave the liquid and go into the gaseous phase, if there's no lid there, they're just going to take off. Less and less molecules in that liquid phase means that there is less energy because they're not colliding as much. Less kinetic energy is directly associated with lower temperatures. And so the solution will actually cool. How much will that solution cool? Not a huge amount, but it is measurable. So evaporation results not only in decreasing the volume, but also in cooling. Vapor pressure of liquids is direct phenomena of evaporation. And also we can see this when we heat a solution up. In a closed container, we can measure the vapor pressure. In a closed container, we have both evaporation, that is, molecules escaping the liquid phase going into the vapor phase, and condensation, that is, molecules that are condensing and being recaptured into the liquid phase. Initially, the rate of evaporation is going to exceed the rate of condensation because in order to get condensation, we of course have to have molecules that are in the vapor phase to come together and be recaptured. The number of vapor molecules is going to increase. That is, evaporation rate is going to increase, which increases the potential collisions with liquid molecules, and therefore we get condensation. Equilibrium is a condition in which opposite processes are taking place simultaneously. So when we measure the vapor pressure, we have an equilibrium between the amounts of molecules that are leaving the liquid to go into the vapor phase and the amounts of molecules that are condensing from the vapor phase to go back into the liquid phase. When we look at a system that's in equilibrium, there is no net change between the amount of vapor we have and the amount of liquid we have. There is just the equal exchange of simultaneous molecules going into the vapor phase and going back into the liquid phase. The vapor pressure is the pressure exerted by a vapor above a liquid when the liquid and the vapor are in equilibrium with each other. And volatile substances evaporate readily at room temperature because they have high vapor pressures. And so that's just a definition of a volatile substance. What you can remember is that volatile substances evaporate readily at room temperature and that they have a high vapor pressure because they evaporate readily at room temperature. Let's look at a picture and try to explain this equilibrium and also this vapor phase because it's a little easier if you're looking at a diagram rather than text. So in panel A on the left here, we took a petri dish and all of those red spheres represent the molecules of the liquid that we put into the petri dish, and then we enclosed it completely. And so this cube that you see is a sealed glass case. Notice here that we haven't reached equilibrium yet. This is before equilibrium. And you can see all of these black arrows that are pointing upwards representing evaporation. So the rate of evaporation is substantially greater than the rate of condensation. And again, as I said on the previous slide, that's because we can't have condensation until these vapor molecules start crashing into each other and recondense into the liquid phase. After some time, you see that we have a lot more molecules in the vapor phase now. And when we reach equilibrium, you can see that we have 1, 2, 3, 4 black arrows pointing up and 1, 2, 3, 4 black arrows pointing down. What that means is that we have an equal rate of evaporation and condensation going on inside of this container simultaneously. So this system is in equilibrium. Now I want to be clear with you, that doesn't mean that we have exactly the same number of molecules in the liquid phase that are in the gaseous phase. That's not equilibrium. What equilibrium means is that we have an equal number of molecules converting into the gaseous phase as the molecules that are recondensing to convert back into the liquid phase. At this point, when we have equilibrium, we can measure the vapor pressure. Now, what is the vapor pressure? All of these gas or vapor particles are going to put some pressure on the surface of the liquid in the Petri dish, because that's what gases do. They form pressure when they're in a closed container. So those gaseous particles are going to form a pressure on the surface of that liquid, and we can measure that pressure that's formed from those gaseous particles. When we get a measurement of that pressure of those particles in a system that's in equilibrium, as this one is, we call that measurement the vapor pressure. And every substance has its own unique vapor pressure associated with it. Just like you learned, every substance has its own unique density. Every substance has its own unique vapor pressure. Every substance also has its own unique melting point and boiling point and sublimation temperature and deposition temperature. So when we look at these two containers, now we looked a minute ago at evaporation, and we had a smaller surface area and a larger surface area of evaporation. Now we're looking at vapor pressure. And on the left, we have a small surface area. On the right, we have a large surface area. And the question is to understand or explain why the vapor pressure is the same for both containers. And we know it's the same because we have the vapor pressure right here pointing into both of these containers, and it says 23.8 torr. Now remember, with evaporation, that occurs at the surface of the liquid, and with vapor pressure, that also occurs at the surface of the liquid. With evaporation, we're looking for the rate at which the particles can leave the liquid and go into the gaseous phase. And so if we have a greater surface area, we're going to have a higher rate of evaporation, because evaporation, in other words, those particles escaping the liquid phase into the gaseous phase, takes place at the surface. But here, let's think about what's happening with vapor pressure. So we have a cap on both of these systems. Initially, we have evaporation going on. Once we have evaporation going on, then those gaseous particles will start colliding and recondensing back into the liquid phase. Where does that happen? That happens at the surface. When we reach equilibrium between those gaseous particles that are colliding to recondense and those liquid particles that are evaporating into the gaseous phase, we measure the pressure on the surface of the liquid, and that pressure that we measure is equal to the vapor pressure. It doesn't matter if we have a very small surface area or a very large surface area. We're going to measure for a given substance the same pressure exerted on the surface, be it a small surface or be it a large surface. We're going to measure the same pressure on the surface. And that's why the vapor pressure is going to be the same, no matter whether you have a small surface or a large surface. Now, let's think about if we increase the volume, for example. So we have a larger volume in the bigger dish on the right. Let's get a bigger test tube that has the same diameter and increase the volume. As long as we still have the same diameter of the test tube, or even if we got a bigger diameter of the test tube and put a larger volume, it wouldn't matter, because the vapor pressure is measured as the pressure at the surface when the liquid particles and the gaseous particles are in equilibrium. So larger volume is not going to alter the vapor pressure. Boiling is the conversion from the liquid state to the vapor state, but this doesn't necessarily occur just at the surface like evaporation does. Boiling occurs within the body of the liquid through formation of bubbles. And so all of the bubbles that are forming inside of a liquid body that's boiling. So say you had a beaker full of water and you put a Bunsen burner under it and you start heating it up. You'd start seeing bubbles forming first around the sides, the inside walls, and the bottom of the beaker. And then those bubbles would get bigger and bigger. Pretty soon you'd be seeing bubbles inside the body of the liquid, and they would be rising towards the top, where the molecules would escape into the gaseous phase. But those bubbles are filled with vapor. That's how they form. You have bubbles, you have particles that are moving away so that the particles inside of the bubble can fill with vaporous molecules. That's what boiling is. Vapor pressure increases as temperature increases. So you can imagine why. Because if you start increasing the temperature by putting a Bunsen burner under a beaker, more molecules are going to be going in the vapor phase. That means that you're going to have more gaseous molecules above the surface of that liquid, putting pressure on the surface of that liquid. At the boiling point, the vapor pressure becomes equivalent to the external pressure that's on the liquid. And in open containers, we can say that external pressure is the atmospheric pressure. In closed containers, we can control the pressure that we're putting on the surface of a liquid. There are, of course, fluctuations in external pressure in the lab if we're working in open containers. Think about altitude changes. And so if we're at a higher altitude, we're going to have a lower pressure. If we're at a lower altitude, we're going to have a higher atmospheric pressure. We need to be able to then normalize our boiling points so that scientists who are at different altitudes in the world can still compare their data. So we say that a boiling point is the normal boiling point at a temperature which the liquid boils under the pressure of 760 millimeters Hg, which is 1 atmosphere. This little diagram is really helpful because it's showing little red arrows where the atmospheric pressure is pushing down on the surface of the liquid. Now, think about being inside that liquid if you were that little gaseous, vaporous bubble that's forming inside the body of the liquid. And of course, we would be seeing plenty of those, not just one if we had a beaker of water over a Bunsen burner. But here, for the sake of having a discussion, we just show one of those vaporous bubbles. What happens is, as the heat heats up the liquid, the particles start to move around. They gain kinetic energy and they start moving around. As those particles move out of the way, this bubble is able to form with a molecule in it that's moving around at very high speeds, or more than one molecule that's moving around inside that bubble at very high speeds. So it's almost like those bubbles are little mini balloons that are being formed in there. As the heat increases, the kinetic energy increases inside of that little bubble, and the molecules inside that little vapor bubble move around faster and faster and faster until that bubble gets bigger and bigger and bigger and has more and more and more pressure inside of it, and it can overcome the atmospheric pressure that is holding it down inside of the body of that liquid. So we say that the boiling point is when the vapor pressure of the bubbles inside that liquid are equal to the atmospheric pressure that's pushing down. That's when we measure a boiling point. Atmospheric pressure impacts this, of course, because if we had less atmospheric pressure, then that bubble would be able to escape the liquid with less pressure inside of it. And we wouldn't need as much heat because we wouldn't need as much kinetic energy. So decreasing the altitude, increasing the pressure, increases the boiling point. External pressure, for example, can be done without an open container. And so we're always talking about atmospheric pressure when we have an open container. If we have a pressure cooker, this is how we cook food really fast. Because in a pressure cooker, we increase the pressure above the surface of either the food or the liquid that the food is in. If we increase the pressure, then the temperature has to be higher in order to get the kinetic energy inside of those bubbles high enough that there's a vapor pressure inside of those bubbles that's high enough that they can escape into the gaseous phase. That means that our food is going to cook at a much higher temperature. Autoclaves that sterilize equipment use the same basic premise. They increase the pressure inside of the autoclave. When you increase the pressure inside of the autoclave, the temperatures that are associated with any objects that are inside of the autoclave are going to heat at a much higher temperature. Liquids with normal boiling points that are particularly high, they're oftentimes in the lab when we need to be able to purify a liquid. And so if we have a liquid that has a really high normal boiling point and we want to purify it, we need to reduce the external pressure. And that way that liquid will boil at a lower temperature and we can do a distillation or a purification at a lower relative temperature. Colligative properties are associated with all of these changes in state of matter that we've been discussing. This is a physical property that's associated with a solution, and it's dependent upon the number of solute particles or the concentration of solute particles. It is not dependent upon the chemical identity because there is no chemical reaction that's going to occur here. Examples of the colligative properties we're going to talk about are vapor pressure lowering, boiling point elevation, freezing point depression, and osmotic pressure. Vapor pressure lowering occurs under a constant temperature, and this colligative property and all colligative properties occur spontaneously. And so something called entropy, or the persistence for a system to move toward disorder, is highly responsible for colligative properties, but we aren't far enough in chemistry to talk about entropy yet. So we're going to look past entropy and talk about colligative properties in terms of other things that you can understand. So this vapor pressure lowering occurs at a constant temperature. We have the addition of a non-volatile solute to a solvent. Now remember, we talked about what it is to be volatile, and so non-volatile is going to be the opposite of that. A volatile substance evaporates readily at room temperature. That means that a non-volatile substance is not going to evaporate readily. So if we add a non-volatile solute to a solvent, that solute is going to stay in the solvent. Solute molecules that stay in the solvent are going to occupy positions not only in the body of the solvent, but also on the surface of that solvent. There are a number of surface occupying solvent molecules, but if those non-volatile solute molecules take their place, now the solvent molecules can't get to the surface as easily. And so not as many solvent molecules can escape into the gaseous phase. Increasing the number of solute particles, non-volatile solute particles that we add to that solvent, is going to correspondingly decrease the vapor pressure more and more. Now, if you think about this, let's just draw a little sketch of what we're talking about here. And so suppose we have a beaker and we have water in that beaker. And then we add to that water a non-volatile solute. We'll make it red. And so that non-volatile solute is going to take up some spaces in this solvent. And again, it's going to take up spaces in the body of the solvent, but it's also going to take up spaces at the surface. If it's taking up spaces at the surface, then as you can see, any particles that are trying to get out, they could get out. So a particle, let's make these round, could get out here. But if there were a particle here, it's bumping into that solute. It can't get to the surface. Particle could get out here into the vapor phase. But if we have particles here or here, they're getting blocked from going from the liquid phase into the gaseous phase. If we have less particles in the gaseous phase, there's going to be less particles putting pressure on the surface of that liquid. And so that's what vapor pressure lowering is. Boiling point elevation, we can draw a similar little diagram of boiling point elevation. And so let's say that we have another beaker. And in this case, with our beaker, we're going to have some sort of flame. I know that's pretty bad flame, but that's the best I can do. You guys can laugh later. And so again, let's say that we had water in here. And maybe if I color the flame red, it'll be a little bit better. I don't know. Okay, let's talk about putting a non-volatile solute into this solution and see what happens to the boiling point. So if we have a non-volatile solute, again, it's going to take up spaces on the surface, and it's going to take up spaces throughout the body of that solvent. In order for a substance of any sort to reach its boiling point, remember, we have pressing down the atmospheric pressure. So the atmospheric pressure is pressing down. We need bubbles of that solvent to be able to get enough kinetic energy that they can break through the surface. And so we're forming bubbles in here, but they're being blocked by that non-volatile solute. Now this one can break through the surface and maybe gain enough kinetic energy to get up out of there. There might be one over here that could squeeze out and get enough kinetic energy to have enough vapor pressure inside of it that it can oppose the atmospheric pressure of the gas pressing down on the surface. But you can see that because the non-volatile solute is blocking a lot of those bubbles from getting up to the surface, and also blocking the ability of movement to go on in this solvent. And so you need movement to increase the kinetic energy. So even though there's heat there, there's not enough kinetic energy, there's not enough movement to get enough vapor pressure inside those bubbles to oppose the atmospheric pressure that's pushing down on them and get out. So what do we have to do? We'd have to increase the heat. So we have to turn up this heat down here. So I'm going to turn up the flame. I know, it's bad. I get it, you guys. We turn up the flame. When we turn up the flame, we elevate the boiling point. That boiling point elevation will then allow those particles to get enough kinetic energy, those bubbles to get enough vapor pressure inside of them that they can escape into the gaseous phase. Decreasing the atmospheric pressure is going to be associated with a decreasing boiling point. And so if we didn't have as much atmospheric pressure up here, suppose we erase a couple of these and our atmospheric pressure decreases. Now, because the atmospheric pressure has decreased, then we don't have to have as much vapor pressure inside of those bubbles because there's not as much pressure opposing them to get out, escape into the gaseous phase. Freezing point depression can also be explained with a similar little drawing. And so with freezing point depression, again, let's say that we have a beaker. And let's say that we have a solvent in that beaker. Let's just have water as our solvent that's in this beaker. And again, we're going to put a non-volatile solute in here. When we put a non-volatile, remember, non-volatile means it doesn't evaporate. It's going to take up all kinds of spaces inside of this solvent, and some of those are going to be surface spaces, and some of those are going to be in the body. Now, we have molecules inside, and if this is water, then we have H2O molecules. And so if we draw some of those H2O molecules, we might have an oxygen here, and a hydrogen here, and a hydrogen here. And we might have an oxygen here, and a hydrogen here, and a hydrogen here. And what has to happen with the attractive forces is the oxygen, which is partially negative, and I can draw some of these over here. As you know, the oxygen, this is polar, and so the oxygen is partially negative, and the hydrogens are partially positive. And so if we had more oxygen molecules, so suppose we have a hydrogen here, and an oxygen here, and a hydrogen here, and then an oxygen, and a hydrogen, and another hydrogen. You notice what's going to happen is they're going to line up. So those partial positive charges and those partial negative charges attract one another. And so we get what we call a hydrogen bond. So there's a hydrogen bond there. There's a hydrogen bond there. There could be a hydrogen bond here. And the water molecules are going to line up such that we have this hydrogen bonding going on. But suppose that we put into the middle of this works a non-volatile solute. That non-volatile solute is going to block that hydrogen bond from forming right here. And because it blocks that hydrogen bond from forming right there, then the water won't as easily be able to form a solid because it can't get that organized structure together that it needs in order to form a solid. It's got to somehow get that non-volatile solute particle out of there. So it's got to push that particle out of there. The way that it's going to push that particle out of there is the water molecules are going to slow down. And the way that they slow down is by spontaneously allowing the system to cool. So the water molecules spontaneously allow this system to cool off by releasing more heat. And as they release more heat, they push that solute particle out of the way. When they push that solute particle out of the way, they can now form a hydrogen bond here. And now they can form a solid by freezing. Another colligative property is osmotic pressure. Now, osmosis is not a colligative property, but osmotic pressure is a colligative property. We need to understand a little bit about osmosis in order to understand osmotic pressure. A lot of you probably know diffusion, and so you probably should write down that diffusion is the movement of particles. Osmosis is the movement of solvent, because that's going to be really helpful in terms of understanding osmotic pressure and osmosis. So with osmosis, we don't have the movement of particles. We can't have the movement of particles because there's a semi-permeable membrane. And that semi-permeable membrane blocks the movement of particles. So the only thing that can move in this system is the solvent because the particles are small enough to move through the semi-permeable membrane. Now we can have different semi-permeable membranes and they allow passage of different sizes of particles, some larger sizes than others. The net flow of the solvent then is going to be from the dilute chamber through the semi-permeable membrane to the more concentrated chamber until the particle concentration reaches equilibrium. And it may not ever reach equilibrium, but that's what it's striving for. And again, this process is spontaneous, this equilibrium process. So the net flow of solvent across the membrane is going to be equal on both sides. Again, that's the net flow of the solvent is equal on both sides. The volume on each side of the membrane, will remain constant when we reach equilibrium. But again, it's very rare that this system is going to actually reach equilibrium, maybe homeostasis in a biological system, because if you're not in equilibrium in a biological system, the cells are going to rupture or the cells are going to shrivel. So let's look at a diagram and talk about osmosis and osmotic pressure. If we look at panel A over here. So in panel A, we have a beaker or a container of some sort that has pure water. That means purely H2O. There are no ions in it. There are no metals in it. There's nothing else, just H2O. We take a tube that has at the base of it this semi-permeable membrane right here. So that's a semi-permeable membrane. It will only allow the passage of solvent, that is water molecules, but nothing else. Inside of that chamber, you see that it's red. This is red, this solution. So inside of that chamber, we have a solution that has a dye in it. And dyes are not macromolecules, but they're pretty close to macromolecules. They're very, very large molecules. And so they cannot pass through that semi-permeable membrane. Only the water molecules can pass through. So there's a whole bunch of huge dye particles or dye molecules inside of that chamber. That's where the red is. And nothing but water molecules, very small, tiny water molecules in the container that's holding everything. So what's going to happen? And again, we can't have diffusion happening. Diffusion being the movement of particles, because those particles, those dye particles, are too big to go through that osmotic membrane. So we can't have diffusion. We have to have then osmosis. And osmosis is the movement of solvent. So which direction is the solvent going to move? It's going to move through the semi-permeable membrane into that same chamber where we have the dye solution. So moves into the chamber where the dye solution is, and what happens? The volume increases. So you can imagine if you were some kind of a little bug sitting up here on the surface. So let's say we had some little bug. Yeah, you guys are going to laugh when you see me draw a little bug. Yeah, some kind of a little bug. I know it looks more like a rabbit, but let's just imagine it's a fly. Sitting on the surface, that fly is going to get a great ride because, you know, it's kind of like Magic Mountain. It's going to go for a ride all the way up to the top here. As the solvent goes into the chamber to try to reach equilibrium. Now, the situation is, what is pushing the bug upward? Why is the bug going for a ride, and what is pushing it upward? And what's pushing it upward is osmotic pressure. That's what's pushing it upward. As the solvent flows into that chamber from a less concentrated to a more solute concentrated region through the semi-permeable membrane, pushes that bug up because the volume increases, and it's pushing it up with what we call osmotic pressure. Now, how do we measure osmotic pressure? We can put a piston and a weight that's equal and opposite to the pressure. Now, how do we know when it's equal and opposite to the pressure? It stops osmosis. And that's what we have over here in panel C. We have this piston and this weight, and we've put pressure on the system until it stopped the process of osmosis, and we can measure that value and determine what the osmotic pressure is in this system. So this slide is just explaining what I just told you, that osmotic pressure can be measured by applied pressures that prevent osmosis. That prevents the net flow of solvent across the semi-permeable membrane. Osmotic pressure and osmosis both are dependent upon the number of solute particles that are present. So you can imagine if we get that chamber more and more concentrated with more and more particles, we're going to have a faster rate of solvent moving into that chamber. This is dependent not only on just adding more particles, but adding more of the right kind of particles. And so if we add, for example, ions, ions in solution separate. And so if, for example, we had magnesium bromide. So magnesium bromide is an ionic compound. It's going to separate into one magnesium with a 2+ and 2 bromide ions with a -1. That's a total of 3 particles that we have then on this side. 3 particles. On the other hand, suppose we had a molecule. Like, for example, suppose that we had something like sucrose. If we had or glucose. If we had something like glucose, this molecule does not separate into ions in solution. And so because it doesn't separate into ions in solution, we have only 1 particle. So adding the right kind of species that will dissociate into multiple particles will impact these colligative properties more so than others. Osmolarity is not something I'm going to have you calculate, and it's not really even something you have to memorize. So this slide isn't there for you to memorize anything on this slide. The only thing that I want to point out to you is that when you're calculating something like osmolarity, this is the molarity, which is the concentration, which we were just talking about. How many particles do you have? Molarity, as you know, is moles per liter. So how many moles do we have per liter is what molarity measures. And we multiply that times I. And this I value represents the number of particles that you have. And so from, for example, 1 mole of a substance. And so you can see that when we're measuring something like osmolarity, this is a trail end of this colligative property we've just been talking about, all of these colligative properties, that the number of particles makes a difference in our calculations. Again, for this class, you're not going to have to do any calculations. You just need to be able to thoroughly explain, understand, and analyze colligative properties and changes in states of matter. These colligative properties, particularly osmotic pressure, are associated with biochemistry because, as I said a little bit earlier, without it, we wouldn't have turbid cells and they would either shrivel or they'd burst, and then people die from things like that. So we have to make sure that we have intracellular fluid and extracellular fluid that results in what we like to see as an isotonic solution. If you put a cell in an isotonic solution, let's say we had a cell and it's in an isotonic solution, and let's just draw a picture of what that might look like. And so that might look like, here's outside and here's inside solute. And you can see that's a pretty even distribution. And so because of that fairly even distribution, that means that the extracellular fluid and the intracellular fluid are going to exchange at the same rate or a very similar rate. And that's what isotonic means. Now, suppose we had a cell in a hypertonic solution. So a cell that's in a hypertonic solution means that we have a lot on the outside. Our solution is hyper, right? Hyper means a lot. Like, have any of you ever seen Hyper Kids? I had a Hyper Kid. He's a lot. He's still a lot. So hypertonic means a lot relative to the inside of this cell. And so if we have a hypertonic solution like this, which direction do you think the flow of solvent's going to be? And remember, this isn't diffusion. The particles aren't going to move. It's going to be the solvent, the intracellular, extracellular fluid. And so in order to get equilibrium, the fluid inside of the cell is going to move out at a higher rate than it would be moving in. Because again, the system is trying to reach equilibrium spontaneously. And so the system is trying to make the extracellular fluid as dilute as the intracellular fluid is, so that they come to equilibrium in concentration. There will be a very small amount going back into the cell, but not very much. And so what happens to a cell like this? It's going to shrivel. And in a red blood cell, that's called crenation. What if, on the other hand, we had a hypotonic solution? So hypotonic solution means that there's not as much inside the cell. Then it's going to be a lot of solute. And so in this case, what direction do you predict will be the flow of solvent in order to reach equilibrium? Extracellular fluid is going to be going in to the cell to try to make it as dilute as it is outside of the cell. And so what's going to happen to a cell like this? It's going to enlarge. And if it enlarges enough, because there's a big enough difference between the solute concentration inside the cell and the solute concentration outside of the cell, then we're going to get what we call hemolysis, where this cell is going to burst. It's going to get large enough to burst. Now, that's not always the case. I mean, we do burst cells in the lab. We will have a hypotonic solution that we put cells in the lab. We want them to burst because we want to pull proteins out of them, or we want to pull DNA out of them, or whatever the case is. But in a biological system, we don't want cells bursting like that. And so oftentimes there'll be an influx of solvent into the cell. And then now you can probably see why we have electrolytes, so many electrolytes, because those electrolytes are ionic compounds. And so those ionic compounds can very quickly reestablish homeostasis because of the number of particles that you can get from them. Dialysis is a type of semi-permeable membrane, because the kidneys use dialysis to remove waste from the blood. If the kidneys are failing, then we need to be able to do something similar, and that similar process is called dialysis, where we have a semi-permeable membrane that allows solvent, and so we literally transfer through a semi-permeable membrane those species that would normally be transferred through the kidneys. And we leave the molecules that are necessary for sustained health, and we remove those waste particles in the process of dialysis. That wraps up this lecture on states of matter and colligative properties. This lecture, I know, is a little bit different from a lot of the lectures you've had so far that are driven by mathematical operations, dimensional analysis, and formulas. And so this is a lecture where you're going to need the flashcards probably more, because you're going to need to be writing things down. One of the best ways to understand these systems is to draw some diagrams. And so if you're drawing diagrams, that'll help you a lot. I've posted a lot of videos to help you through these colligative properties that have visuals and simulations that you can understand. Even though I posted a lot of videos, they're not long videos. So it isn't going to take you a lot of extra. Some of these videos are a couple minutes, 2-3 minutes. So it isn't going to take you a lot of extra time, but it'll give you some good visuals, and that'll help you to be able to draw some sketches of your own so that you can explain these changes in states of matter and also the colligative properties, because obviously the test questions are going to be multiple choice test questions, the likes of what you have, the type you've seen in the past on exams when I'm asking about conceptual information.